Thank you very much, Pete, for that great brief overview of all the exciting research that's going on at Edinburgh. Unfortunately, Pete isn't able to join us now for the live Q&A session with our speakers. So can I ask all those speakers that are able to come on screen to come on screen now, and I'll try and get through as many of the questions that have come in as I can, and also give you the opportunity to ask each other questions, because I'm sure you'll have some. To begin with, there have been quite a few questions coming in about parasitic plants. I think, and with Charles's at the end on Rafflesia. So I might just kick off with a few of those, if that's all right. There was one of many, and important for botanic gardens. Are any Rafflesiaceae grown in botanic gardens around the world, e.g. in Southeast Asia? So perhaps that one's for you, Charles, and then maybe Chris, you'd like to comment.
I was going to punt to Chris actually, but the only instances I know about this are in the Bogor Botanic Garden in Indonesia. It's a smaller flowered Rafflesia that they've had growing there within the garden grounds.
Yeah, I'd concur with what Charles says. The only botanic garden example is in Bogor in Indonesia, which is sort of falls within the natural range of Rafflesia. There are some unusual isolated examples of people in Sarawak, I believe, and Malaysian state of Borneo, who have actually managed independently of a botanic garden, as I understand, to grow Rafflesia in a sort of small holding actively, sort of almost in their back garden, which is quite extraordinary. But it's certainly not widespread in botanic garden collections by any means.
No, and I should mention that picture I showed of you, Chris, at Oxford was not with a Rafflesia that we'd grown. It was of a rather extraordinary life-size model that
It did raise my eyebrows, Simon, because I didn't know you were going to show that.
Yes, well, that's a point to aspiration for
very aspirational
botanic gardens to try and grow some of these ultra challenging plants among our collections. There are a couple of others also on Rafflesia and the like before we move on. This is a good one. Are different species of Rafflesia hosted by different species of tetrastigma? Perhaps Charles.
Yeah, so they're quite specific in many instances. So there's a relationship where there's one host species and one parasite, but a handful of them actually can parasitize more than a single species of tetrastigma. So there's some variation across the group. What I would add though is that the taxonomy of tetrastigma is sort of poorly known and understood, and this is an area where museum collections obviously help us. But one of the great challenges of these host species is that they're in many cases large woody lianas that shoot from the ground and up into the canopy. So often, obtaining the flowering and fruiting material that's necessary to delineate species, they're not terribly well represented in herbaria. So I think actually, the relationship is very narrow. With this one-to-one relationship, and some species that parasitize more than one tetrastigma species, sometimes of distantly related tetrastigmas species, but I would guess that there's surprises yet to come as we learn more about the sort of taxonomy and diversity within the host genus tetrastigma.
Okay, thank you, Charles. Now moving on to Alex, there's a question about your fascinating talk, and where do you think your conservation research findings are having or will have the most practical impact?
Thank you. That's a very good question, of course. And to some extent, it's very reliant on data and data gathering, as I was trying to make the point. In many countries that Kew is working with, they have not yet established priority areas for conservation. So we are running a program at Kew called the Tropical Important Plant Areas, which has been running for a few years now, I believe in about 7 countries. And we've worked with partners across Latin America, Africa, Southeast Asia, and identified about 144 areas for prioritization for conservation in those countries. So the hotspots of plant diversity, so to speak. And this is a work that involves a lot of stakeholder engagement. And we're hoping now that this methodology will be able to speed that up and improve some of that classification as well. So that we are absolutely confident that when countries decide which areas to conserve, it's absolutely the best ones. And I believe that this is really the most important application area for this research.
Great, thank you, Alex. And there's a question now for you, Barbara, going back to the beginning. This trigger of ovule and seed coat development after pollination, do you know anything of the nature of the cues that, the signals, the cues that come from the pollination event that then trigger that development?
For the moment, we have no clear idea. In the pollination drop, there is some metabolites that are involved, but we think that the pollen grain, when is inside the ovule, grimp and stay attached to the gametophyte, female gametophyte, and stay for about 3-4 months before is able to germinate. And so we are looking for the trigger for this development. We see that the genes some genes are regulated to form the integument, seed integument. And so the seed integument is formed before the fertilization occurs. So this is quite triggering because usually you can talk about seed coat when you have the embryos inside, not before. So we are looking for the triggers for the moment.
Fascinating system in that effectively living fossil.
Yeah.
Thank you. And now coming over to you, Max, a lot of people have put likes in for stinging nettles. Amazing this sort of sexual gender diversity among stinging nettles across the world. Is there any ecological, do you know of any yet ecological correlations between the different sexual systems that we see in stinging nettles? For instance, androdiaceae among some plants seems to be associated with particularly poor soil types and stressful environments. Have you got any correlations in nettles?
No, we've actually got none at all. The interesting phenomenon is that Urtica stinging nettles, they always grow in exactly the same type of microhabitat. No matter where you look at, they grow in fertile cave mouths in New Zealand. They grow in fertile moist patches in Ethiopia. They grow in the very same places in Europe. So they are ecologically, they've got virtually zero variability. The only phenomenon is that you get in highly seasonal habitats, you get annual ones, and in less seasonal ones, you get perennial ones. And that doesn't even correlate with phylogeny very much. There's a lot of shifts in life history going on within the phylogeny. As a matter of fact, we've got absolutely no explanation for the different gender distributions that we find in Urtica. The only exception is that the annual ones tend to be, well, they tend to be monoecious, very, very carefully wording that. They tend to be monoecious, but we've got a very strong indication that ontogeny plays a major role in the gynodioecious and andromonoecious species, that if they are very well fed, they've got a lot of water, a lot of nutrients, they tend to become monoecious individuals. And if they are somehow starved, if they're very crowded, you get unisexual individuals. We never went in, we worked with 50 species in cultivation. We never went into large-scale studies on individual species. So I think there's a lot of ontogenetic regulation also going on with regards to monoecy versus unisexuality, but the gender distribution on the individual plants is highly conserved. So it's within the individual species. So where the male and the female flowers are ultimately formed, if both are formed, is highly conserved. But whether both of them are formed shows some degree of variability. I've been thinking a lot about any adaptive reason behind the different geometrical distributions of male and female flowers, and I'll welcome any suggestions. I can't come up with any. I've got no clue why they do it. They just do it because they can, I guess.
Extraordinary. Right. Now, we haven't got that much longer for you answering questions, and we've got more questions than we're able to fit in answers for. There have been a few come in to do with horizontal gene transfer that I could, Charles and Chris maybe comment on these and others chip in. There's one about how does horizontal gene transfer affect speciation? And there's another one about whether it's possible between insects and plants, I suppose aphids and other intimate herbivores. Charles, do you want to kick off there?
Yeah, I'll start with where you left off. Actually, back in March, there was quite an exciting paper in Cell that was published that demonstrated that a white fly, an agricultural pest, that acquired a gene from plants. So, quite a long evolutionary distance in terms of gene transfer. And in this case, this gene allowed the insect to basically counteract the defenses of the plant. So really pretty extraordinary. And I can post a link to that paper in the chat window. So I think, early on when we started exploring gene transfers in eukaryotes especially, there was thinking that this wouldn't be common. And increasingly, as we have more and more genomes, it appears to be quite common. The extent to which it facilitates speciation, at least at macroevolutionary timescales, is still a bit of a mystery, but almost certainly, I would wager that's likely to be the case, and certainly this example of this agricultural insect pest is one, I think, very nice example of that.
Thank you. I guess you don't want to add any more to that, Chris.
No, I don't think I could, only to say from a broader perspective that clearly host parasites are these fascinating living laboratories for exploring questions such as these and these sort of elevated levels of communication between host and parasite. So,
right. So I think perhaps we'll start to, well, why don't, if you have any questions to ask other members of the panel, we've got a few minutes left, perhaps time for one or two questions. No. Matt.
Yeah, actually, yes, I've got a question for Chris, because we've been looking at the emissions from Araceae, from Mediterranean Araceae as well, and we are having a major headache with the ammonia emissions. They're messing up all our measurements because, well, usually if you use an adsorbent, it's not absorbed. So you won't even see it in your analyses maybe. But with the GCIMS, you get everything that the plant emits. And ammonia emissions are just swamping every other signal. Do you have any take on that? Because it seems to be ecologically highly relevant, ammonia being such an important molecule in plant-animal communication.
Yes, so the chemist with whom I work could give you a more informed answer. What I will say is I share your pain, Max, because for nearly 4 years, I think, we've been trying to optimize our methodology. And actually, sometimes we've lost a whole year's data because I'm sure you'll have the same problem. You've only got a limited window to samples. This is the messy side that we don't present on our slides. And even the particular plastic that we were using to enclose the inflorescences with the probes on which we were absorbing the compounds to later desorb them in the lab, that ruined a whole year's worth of data. So yes, what I can say is that the probes we're using specifically are designed to pick up the sulfides, so disulfides, trisulfides, oligosulfides, that kind of thing. And so I think it's fair to say that in an ideal world, we would be looking at lots of different classes of these volatiles that are produced, and we're not for logistical reasons. But so that sort of partly answers your question, I think, but we haven't had that problem specifically. We've had many others. But maybe we could have another chat offline and to get into some of the detail and share one another's learning, so to speak, from this very difficult and delicate process.
Yeah, let's do that. Thanks.
Okay, well, we're victims of time, I'm afraid. So thank you all for joining this session, and thank you all for your talks and contributing to this this symposium. It's been a great first session. I wish we could all clap, but let's all participants give a virtual clap for our speakers this afternoon.